Hey there, everyone. Welcome back to our another video. We are going to do viral replication today. So we're going to talk about viruses in their replication cycle. We had another video a little bit earlier about the viral tutorial, what makes up a virus. So this time we're going through the replication of a virus. So a viral replication cycle is going to have 5 primary steps. The essential portions of this are going to be as follows. We're going to have infiltration. So the virion is going to be able to access the cell somehow, right? It is going to then be able to be transported to the nucleus. It is going to invade the nucleus. It is going to transcribe itself into the nuclear genome of the host cell. Once it does this, it will have messenger mRNA production that will shoot that signal out to the ribosomes, which will then translate that viral replication and that viral genome. The viral proteins will be continually made, and then a new virus will bud off from the cell. So, what are the details of this process? All right. So, we've got an active virus life cycle here. And that active life cycle is going to be everything that is constituting infection all the way to leaving the host cell. So, our 5 steps are attachment. Virion has to attach to a host. Second is penetration. It has to infiltrate into the host, and it has to find its way to the nucleus. 3 is synthesis, right? It has to invade the host genome and begin to replicate itself. So the viral proteins and the nucleic acids have to be copied using the host cell's cellular machinery. Then there is assembly. We have to put those viral proteins together, and we have to create a new encapsulated virus, the viral components. 5 is release. The newly formed virion and virus has to be released from the host cell. So we've got attachment. In this case, we have a bacteriophage in this picture, right? Bacteriophage is specifically designed to inject its nucleic material into a bacteria. We have attached to the surface. Secondly, we have attached and we have now injected. That is penetrating into the cell. The DNA of that cell is going to mix with the viral DNA. We're going to transcribe that, and this is going to be known as phage DNA. The phage mRNA is what is going to be transcribed from that and is sent to the ribosomes for processing, right? Then at the ribosome, we are going to have those phage proteins be produced. So the phage proteins, other components in DNA are going to be produced. The host DNA is degraded and disintegrated. And then finally, our last step is going to be assembly and then release. So we put the phage components together into newly formed virions, and then those virions are released from the host cell. So our first step, attachment, right? Viruses are going to be inert and metabolically inactive. This is why the argument is there that viruses are not technically alive. They have to have a host cell in order to hijack its machinery to reproduce themselves. So a virion or a virus is going to exist independently of a host cell, and it has to be able to penetrate into a living organism's cells in order to get its own DNA to be replicated. So the encounter of a virion to an appropriate host is going to be a random event, right? The attachment, however, the actual latching on, is going to be very specific. So the molecules of the virus are going to locate receptors on the host cell surface, mostly proteins, and those are going to be latched onto and specifically targeted by the viral molecule. After a virus has encountered a host cell that it is viably attached to, now it has to somehow get its nucleic acids into the host by penetrating the cell membrane, right? So what we have here is the process termed penetration or viral entry, okay? One of the times that we see this most commonly is with the bacteriophage. These are viruses that are specifically designed to infiltrate bacteria. An unenveloped eukaryotic virus one that's going to target plant and animal cells, is going to gain entry via endocytosis. Okay, so with the case of a bacteriophage, we had the injection of viral DNA into the host cell via penetration through the cell membrane. In eukaryotic viruses, we're not going to have direct penetration and porage or pore entry into the host cell. We are going to have endocytosis, where the host cell is tricked into actually reaching out and engulfing the viral particle, right? So the host is going to physically reach out and engulf the capsid. And that is what we see in most animals and in eukaryotic cellular hijacking. So an unenveloped eukaryotic virus is going to gain its entry that way. An enveloped eukaryotic virus may gain entry from the nucleocapsid, right? That might cause viral envelope fusion. So we could see unenveloped viruses come in like we would be integrating other particles by phagocytosis and endocytotic vesicles. With an enveloped eukaryotic virus, we might see integration of that envelope via fusion, right? And fusion might happen where the viral envelope is going to fuse with the cell membrane of the host cell, and the nucleocapsid is going to insert its DNA through that fusion point. Regardless of which way we actually gain access into the host, we have to be able to start replication. So the synthesis stage is going to be the most important process that happens here, right? Synthesis stage dictates whether or not we actually have a successful infection. So the viral DNA is going to dictate this process. The specific enzymes for the virus, such as the RNA-dependent RNA polymerases, are going to be necessary for the replication process. So first, we're going to have protein processing, right, and protein production. We have to be able to make an mRNA transcript in order to get those viral proteins to be produced. So to ensure the components are made at the right time in development, the viral genome has to integrate itself at the proper destination at the appropriate time. Next is assembly. The complexity is going to depend on the virus being made. The more complex the virus, the more intricate and tricky and difficult assembly might be. The simplest viruses have 3 components, right? They have a capsid. They have a capsid, which has 3 proteins and self-assembles. All right, so they have 3 protein components. The most complex viruses can have over 60 different protein components. So the simplest, we're going to have 3 components of proteins. Most complex, we have over 60. Every single one of them has to come together in a specific order. So it doesn't matter if you have 20 proteins or 60 proteins. They have to be assembled in a stepwise function. Lastly, after we have assembly, we're going to have to release it. So in order to release, we're going to have 2 different mechanisms. We are either going to lyse the host cell by physically destroying it, killing it, and having the virions be released from the inside of the cell out to the environment by destruction of the host cell, or we can have what is common for envelope viruses, and that is budding, right? Where we see an exocytosis type of vesicle release into the outside environment, where we don't typically destroy the host cell or it doesn't die, but we do see release. So a virus that is going to be infecting a bacteria is known as a bacteriophage. We have a transmission electroscopy here that is showing bacteriophage activity. It's going to latch on to the cell surface, physically inject its nucleic acids into the cytoplasm of the bacteria via the bacterial shuttle. A virulent phage is one that always lyses the host cell. So if it's a virulent phage, that means that at the end of replication, it is always going to physically destroy the host cell. This is called the lytic cycle, right? Where the host cell is lysed or destroyed at the end of the replication is going to be the lytic cycle. because we're always ending in lysis. So we have a couple of options here, right? Option one for a virus is going to mimic a virulent stage, follows the 5 steps of replication, and lyses the host cell in the lytic cycle. All right. There's also temperate phages. These are viruses that have 2 options for replication. Okay. Exposure of a host cell to stressful conditions can also cause induction of a virulent cycle. And the event triggers the remaining steps of the lytic cycle: synthesis, maturation, and release. Our second option for a temperate phage is to remain within the host cell without destroying it. So this is not called a lytic cycle. This is known as lysogeny. Okay. Lysogeny is where we have replication of a virus inside of a host cell, but we don't have destruction of that host cell. So the infected bacterium is known as what is called a lysogen. In this state, the virus is going to have a stable relationship with the host. Okay. So we still have the first 2 steps that occur here. We have attachment and penetration. But after attachment and penetration, we have the replication process happening, right? And it is going to be in conjunction with the host cell. So we don't actually destroy the host cell. So here's our 2 different phases. If we have a full lytic phase, full lytic cycle, then we have no storage or no quiescence or no hibernation of virus. We start out with the bacteriophage. The host bacteria is going to be inserted with the bacteriophage genome. The phage DNA is going to circulate and move its way into the host DNA, right? During replication, the phage DNA is going to be replicated, phage proteins are made, phage particles are assembled, and if it is at the end of the lytic phase, those assembled virions are going to be released from the host cell when the cell lyses and is destroyed. With the lysogenic cycle, we start out much the same, right? The phage infects the cell. The phage DNA is actually going to become incorporated into the host genome. So the cell DNA is going to incorporate the viral DNA into its genomic structure and will replicate it. The cell divides, and what we call prophage DNA is passed on to daughter cells. Eventually, at time of induction, induction can happen by different mechanisms. One that we labeled before was stressful conditions. At the time of induction, the phage DNA is going to be removed and excised from the host genome, host DNA, and then we can enter the lytic cycle. So lysogeny can go on for days, weeks, months, even years, until a stressful event occurs. Something causes that viral genome to separate from the host DNA, and then we enter into the lytic cycle where we replicate the virions and release them after we destroy the host cell. So the details on these 2 processes: lysogeny. Lysogens are going to experience a benefit from lysogeny because it can result in lysogenic conversion. That means the development of a prophage leads to the change in a host phenotype. One of the best examples of this is the Corynebacterium diphtheriae, which is the causative agent of diphtheria. For temperate phage replication, what dictates whether we use lytic cycle or the lysogenic cycle? And this is just based on environment. This is based on the need of a viral genome to survive. So if it is a favorable environment, it does not matter. If it is an unfavorable environment, then the lysogeny might occur. So if there are plenty of host cells around, there's plenty of places that we can hijack cellular machinery, then the virus knows that it has no limit and options of where it can go. So it's likely that a temperate phage will engage in the lytic cycle immediately because it is going to be able to find host cells immediately around it after replication to continue replicating. So this is going to lead to a large increase in viral replication. And host cells that are going to be independent All right. If we don't have any other places around that the temperate phage can go to continue replicating, it's more likely that the host and the temperate phage will enter a lysogenic relationship. This will allow for the virus to survive within the host cell until the host cell numbers increase enough to where we can start to infect a large portion of host cells and reproduce the virus on a large scale. All right, everyone. Make sure that you like and subscribe to the channel, Twisted Science, also the Patreon, patreon.com/Twisted Science for additional videos. Thank you all for watching. I will see you next time. Peace out.